Our next speaker is Dr. Edouard Timsit from the University of Calgary, and he's assistant professor in cattle health, diplomate of the European College of Bovine Health Management, and he will speak to us about the evolution of the nasopharyngeal microbiota of beef cattle.
Thank you very much, Mr. Chairman. So after diarrhea and mastitis, let's talk about BRD now. So first of all, I would like to thank the scientific committee for inviting me here to speak and present my research on the nasal microbiota of beef cattle from weaning to 40 days after arrival at a feedlot. So as you know, bacterial pathogens such as Mannheimia hemolytica, Pastura multocida, H. somni, and Mycoplasma bovis can reside in the upper respiratory tract of healthy animals as commensals. However, under specific conditions, namely stress, viral infection, and suboptimal environment, those bacteria can also explosively proliferate and then can access to the lungs, leading to bacterial bronchopneumonia. The host immunity is very good at controlling the proliferation of bacterial pathogens in the upper respiratory tract. However, there is also recent research showing that the nasopharyngeal microbiota can also play a very important role in preventing or predisposing cattle to bacterial pneumonia. Actually, commensals and bacterial pathogens are constantly interacting in the upper respiratory tract. And as you can see on this graph, commensal can actually either, commensal and pathogens compete for nutrient, adhesion sites, but also commensal can directly inhibit or kill pathogens. And furthermore, those commensal have anti-inflammatory effect or immunomodular effect that can either predispose or prevent the colonization by pathogens in the upper respiratory tract of cattle. So when this flora is at equilibrium between commensal and pathogens, usually it's pretty difficult for the pathogens to colonize and proliferate. However, when you've got a disrupted equilibrium between the pathogens and the commensal, at that time the pathogens can colonize, proliferate, leading to a disease. For example, in human, a disruption of the microbiota following an antibiotic treatment, so here the microbiota, disruption here with the antibiotic treatment, was associated with a temporary increase in the pathogen colonization and overgrowth in the upper respiratory tract, which lead to high risk of developing otitis media. So we know that does occur in human. Unfortunately, because most of the studies actually are focused on pathogen only, we don't know neither the composition of the nasopharyngeal flora of cattle and its role in maintaining or precipitating disease. This is why we did this study, to describe the composition of the nasopharyngeal flora of beef cattle and its evolution from weaning to 40 days post-arrival at the feedlot, which is the highest risk period for BRD. For this objective, we use a metagenomic approach, use the 16S rRNA sequencing to describe the overall composition of the nasopharyngeal flora. So we follow 13 Angus steers, and they all originated from one cow-calf herd. They were all dewormed and castrated at least 45 days prior to the beginning of the study, and they were fully vaccinated against Mannheimia hemolytica and H. somni. They also were vaccinated against all the common viruses for BRD. So at the cow-calf ranch, as soon as the steer was removed from the dam, we immediately sampled them by deep nasal swab, so that was guarded swab. Then the animal was transported for 2 hours in a commercial auction market located in southern Alberta and held overnight in contact with other cattle at this auction market. Then cattle was transported 8 hours to a research facility and offloaded from the truck and allowed to rest overnight. On the next day, the processing day, this cattle was dewormed and resampled by a deep nasal swab. They did not receive any vaccine, they did not receive any antibiotic on arrival. And then we followed those animals for 40 days, an animal with visual BRD signs, and a rectal temperature above 40 degrees Celsius was considered as BRD affected and treated for this condition. Finally, at the end of the study period, D42, we did another nasal swabs for the microbiota. So we really have those three sampling points: weaning, arrival, and day 40 after arrival, so day 42 in the study. So the total DNA was extracted from each nasal swab using a DNA tissue kit. Then, the V3 region of the 16S RNA gene was amplified using specific primers. These 16S RNA gene implicants were sequenced using a MiSeq from Illumina. The DNA sequence was clustered into operational taxonomic units, also called OTUs, using a custom pipeline based on UPORs. Then, the taxonomy was attributed using the Green Gene database. All the downstream analyses were performed in R, and we used the Bray-Curtis deciliary index to create a sample-wise distance matrix to study the beta diversity, also called within sample diversity. Then we used the effect of time, so on the microbiota, and L status, so SIG versus LC, were analyzed using a PermaNova, which is a permutational multivariate analysis of variance. With regard to alpha diversity, which is the intra-sample diversity within the observed species and the Shannon diversity index. So the observed species relates to the total different numbers of OTUs within a sample, whereas the Shannon diversity index relates to the total number of different OTUs and the evenness of their repartition within a sample. So it's two different measures of within sample diversity. So let's move to the results now and the characterization of the nasopharyngeal flora. So in total, we've got 10,500,000 sequenced obtained across all samples, with an average coverage of 100,011 here. 111,000 sequences per sample, so a very deep coverage. The sequences were clustered into 1,342 OTUs, including singleton. So we found 16 different phyla. However, as you can see on this graph, 88% of the abundance actually here were within Ternicutes, 53%, and the Proteobacteria. The other phyla that were more than 1% were the Firmicutes, the Bacteroidetes, Actinobacteria, etc., and other OTUs that were less frequent. If you look into the Ternicutes, 99.8% of the Ternicutes were attributed to a single genus, Mycoplasma. And within those Mycoplasma, 78.5 was Mycoplasma dispar and 4.5 was Mycoplasma bovrieni. Concerning now the Proteobacteria, this phylum comprised Pasteurella, 32%, Rastonella, Moraxella, and Mannheimia, only 2.3%. Among Pasteurella and Mannheimia, they were all Pasteurella multocida and Mannheimia hemolytica. The Firmicutes, we found Penibacillus, Bacillus, and Leptococcus. So I'm not describing the other one because they were less frequent. So now, let's see about the beta diversity and the evolution of the flora. So as you can see on this graph, so just to describe what it is, each dot represents the microbial structure on a nasal swab. So the more distance you've got between two dots, the more different the two flora are based on the Bray-Curtis deciliary index. So the red dots here are for the nasal swab performed at weaning, the blue dots are for the nasal swab performed on arrival, and the green dots are for the nasal swab performed 40 days post-arrival. So as you can see, actually the sample cluster by time of sampling here, not by animal, for example. And by using the PermaNova, we showed that actually time was a significant source of variation, explaining 20% of the total variation, which is very high in terms of microbiota. So in total, 92 OTUs change over time. So it's a very busy slides, just to show you that here is this top 25 OTUs sorted by p-value. And what you can see, there is some OTUs that were actually decreasing from weaning to 40 days here, like for example, Pasteurella. Some other that actually increase, like Rhinolobacillus, that are associated with rumen fermentation, and some other that actually increase and then decrease. So a huge variation in those different OTUs. With regard to alpha diversity, however, we didn't find any difference between the different sample points. So you can see here the observed diversity. all the Shannon diversity, so no difference between those different time points, which means that we had the same amount of different OTUs per time points and an evenness of repartition that was similar between the different time points, so not a lower diversity on arrival as we were expecting initially. Else data now. 3 out of the 30 animals were diagnosed with bovine respiratory disease during the study period. 2 animals were actually diagnosed on day 2, so number 17 and 24, and 1 was diagnosed on day 26, the number 28. So what you can see on this graph, actually, that those animals were clustering with the other one. They were not clustering apart. And based on the Permanova, the health status was not actually also a significant source of variation between those animals. So with this study, we showed for the first time that actually the microbiota of cattle, the nasopharyngeal microbiota of cattle, can undergo a very rapid and profound evolution from weaning to arrival at the feedlot and from arrival to day 40 at the feedlot. And this without any treatment or vaccination. And this strong evolution in the flora can explain why cattle actually are more susceptible to be early in the feeding period. As I showed you in human, we showed that a disrupted nasopharyngeal microbiota actually is less resistant to the colonization by pathogenic bacteria and is also less resistant to pathogen overgrowth. Furthermore, what we showed in this study is that actually mycoplasma, especially Mycoplasma dispar and bovirini, was very abundant in the nasopharyngeal flora, at least in this cattle population. And based on those data, we infer that mycoplasma can be actually part of the core nasopharyngeal microbiota of healthy cattle. So thank you very much for your attention. I would like to thank the Alberta Livestock and Meat Agency for their financial support, as well as the person that worked on that project with me. So Dr. Workentime, that did most of the bioinformatic analysis, Dr. Vandermeer, that is with us today, Dr. Schreiber, and Dr. Alexander. This paper was recently published in Vet Microbiology, I think 2 months ago. And this is my coordinates if you want me to send you a reprint because it's not in open access. So thank you very much for your attention.
Thank you very much, Edouard. Do we have any questions? I see a number of hands.
Thank you very much. I have had an opinion for a long period of time in feedlot cattle that mycoplasmas are the hidden danger for everybody. I would appreciate your opinion on how serious pathogen is Mycoplasma bovis, and did you find any evidence of Mycoplasma bovis as a resident in the pharyngeal area? Thank you.
So, thank you very much for this question. Mycoplasma bovis is a pathogen. There's no doubt. We found it in Canada in numerous cases of necrotic bronchopneumonia. In this study, though, I didn't mention, but it was pretty low. We found less than 1% of cattle with Mycoplasma bovis, so it was really a very low prevalence. My understanding is from all the publication that Mycoplasma SPP, so either bovirini, dispar, are very prevalent and everywhere. However, bovis is way less common, and if you look at most of the publication, they will find up to 80% of the cattle positive for Mycoplasma SPP, but when they do a PCR and they know the species, actually, bovis is less than 1% or 2%. So I would say that in cow-calf herd, when you don't have Mycoplasma, you can have actually a free herd of Mycoplasma bovis. However, in feedlots, because you're putting some cattle from everywhere, usually what we've got is a low prevalence on arrival, and after 40 days, all cattle are positive for Mycoplasma bovis because they spread like very, very fast. So to respond to your question, it's a pathogenic bacteria. Is it part of the resident flora? Not necessarily. It can be part of it, and it can be really prevalent in feedlots. But it's not because you isolate Mycoplasma from the upper respiratory tract that you will have necessarily a bacterial pneumonia due to this bacterium, because we know that for Mycoplasma also the agreement between nasal swab and the lungs is not good.
There is another question right there.
Oh, thank you. I just wondered, is there Do you know, is there any normal expected change in the microbiota over time if the animal wasn't subject to any stress? Or is it expected the microbiota at pre-weaning would stay the same if we didn't do those things?
So that's a very good question, and actually that's one of the other limitation of this study is we wanted to follow those calves before weaning, but the farmers said no. So I would say that in human we know that the microbiota is sort of quite stable over a short period of time without any stress and viral infection, but can change really quickly. One of my key message here is be careful when you study the microbiota because it can change with any stress or viral infection, where we know that it's already changing with antibiotic, but it's not only antibiotic. So, but that's a very good point. And we need to have more data on exactly how stable it is over time and how it can really influence the microbiota. Now we show the change, now we have to explain those changes. But thank you for your question.
Well, thank you very much, Edouard.